My name is Michaela. I am the Bio 190 class, and this is the laboratory assignment for this week. I did a detailed drawing of the IP3 signaling pathway. The black ring around it is the cell, and then everything that happens underneath it in order. So starting at the messengers like that are extracellular signaling molecules, which are hormones and neurotransmitters, will get recognized by the cell. Then the cell will release second messengers, which is IP3, DAG, and calcium ion. With the increase of the cytoplasmic CA2 levels is caused by the binding of hormones and signal molecules, which sends important signals to perform functions that CA2 might need to do. So then when we go down to number 3, this is the endoplasmic reticulum with the CA2s inside of it. There is the CAMP, which is represented right here. It opens calcium channels and plasma membrane for CA2 to rush in. Some CA2s will be replaced, and that is what that is for, is it will open that channel, and the remaining CA2s will go back in there and replace the ones that were lost. When we go up to 4, this up here, the big green circle right here is the G protein. This activates the G protein ATP. It changes from ADP to ATP. This activates the phospholipase and then the phospholipase GQA, and it becomes which is represented right down here, and then it turns into IP2, IP3, and DAG. So IP3 is hydrophilic, which means that it just fuses into the cell. The DAG is a lipophilic, which means it remains in the cell membrane. And then, especially if it's in muscles, the I3P binds to the calcium channels on the endoplasmic reticulum and allows the release of calcium into it. The DAG uses the calcium ions, which is up on here, and it uses it to activate protein kinase C. Once phospholipids are added to the targeted proteins, this causes the final stage of cellular responses. So this is the full diagram that I drew that represents what happens in the I3P or IP3 signaling pathway, step by step. Most of it has to do with a lot of the calcium ones that the calcium ions that are introduced at the beginning, that follows down through, gets replaced in 3, and then this is up here where the proteins get broken down and then comes into contact with the protein kinase C, and then it creates a cellular response.